Okay, good morning, everyone. Sorry, I'm off to a little bit of a late start. I have my Bio 275 lecture right before this. And today we were talking about transcription and translation. And I was actually really happy at the end of the lecture. There were lots of really good questions from students. So, for those of you who take my class, today's the day that I did the whole cookie recipe thing and my story about the Mrs. Fields cookie thing. So I think that gets them kind of into it. So I told them, okay, I'll stick around and answer your questions. But then at a certain point, it's like, sorry, I got to go. Okay, so let's continue our discussion about oncogenes. And we'll talk about RAS in a little bit more detail. We'll look at Src, because we already mentioned Src as a potential oncogene, and we'll look at Myc. Okay. Last time we left off discussing RAS. So we said RAS is a very important proto-oncogene. So our genome has 3 copies of RAS, one on chromosome 1, one on 11, one from 12. And we call these NRAS, HRAS, and KRAS based upon what disease was the basis of them originally being identified as being the important gene. I said that these 3 copies of RAS over evolutionary time have, through millions of years of evolution, have each sort of taken on their own sets of different mutations and polymorphisms and things like that. That means that they are slightly different now from each other. And that's why one of them is sort of more functional in one type of disease pathway versus the other in another. Okay, but they're pretty identical. Last time I ended with this point that nearly all, around 25% of all cancers involve some kind of a RAS mutation, right? And in some types of cancers, upwards of like 90% of that type of cancer, for example, pancreatic cancer, you see something like certain RAS mutations. We said that if you were to look at where these mutations happen within the gene to cause, let's say, an amino acid substitution in the eventual translated protein. If we were to lay out the protein, it's 189 amino acids. So here's the N-terminal end, and here's the C-terminal end of the protein. And you were to map out like 1,000 random tumor samples that you get, and you look for RAS mutations. The shocking thing, right, is that nearly all of them have a mutation right at amino acid 12. And then a few of them have one at amino acid 13. Fewer of them have mutations at amino acid 61, right? And I said, wait a minute, mutations are random, right? Mutations happen randomly. So how could it be, right, that the overwhelming majority of mutations are affecting amino acid 12, right? I asked you to kind of think about this last time, and do any of you have any thoughts as to why you see this? You can unmute yourself, or I can even turn on the chat. Let me see if I can still turn on. How do I turn on the chat if I'm sharing the screen? My chat doesn't even show up anymore. But if any of you have an idea about this, you can feel free to speak up. Right. Oh, okay, I got the chat working now. So if you're shy and you don't want to speak up, you can type in your response.
Could it be because with the mutation, it has an overreplication, it has a selective pressure towards that type of mutation?
Yeah, excellent. That's exactly it, right? So I think I say that on the slide here. The RAS mutations that lead to cancer are the ones that are selectively seen here. Why? Because you've already biased what you're looking at, right? Because you're starting with 10,000 tumors and you're saying in these 10,000 tumors, what are the mutations that kind of led to these being tumors, right? If you took 10,000 normal samples of normal tissue, yeah, you bet some of them you'd find RAS mutations, right? But you wouldn't find this concentration. So it's kind of like, I think of it this way. I think many of you are pre-med, right? And you're going to take the MCAT if you haven't taken it already. Let's say you go down to Ala Moana, right? And Ala Moana, and let's say it's a pre-pandemic day at Ala Moana, so there's thousands of people there, and you randomly sampled 100 people that just passed by, right? And you said, have you taken the MCAT, right? Maybe out of 100 random people, maybe like what, I don't know, 5 out of 100 would have taken it, right? But if you go to JABSOM and you take 100 first-year med students and you say, how many of you took the MCAT, right? It would be 100%. So that's what's happening, right? You're biasing what you're seeing here. But that doesn't mean somehow this is not legitimate, because we're not saying, right? The question is not where in the gene is there possibly a RAS mutation, because it could be any base pair in the gene that gets a mutation. What we're saying is, if you just look at tumors, what evidence can you see that certain amino acid positions in the RAS protein are the ones, when they are mutated, contribute to the cell becoming this tumor cell that you ended up looking at? All right. So let's look at this. What is RAS? Why is it so important as a oncogene? So RAS is what we call a G protein, right? So a G protein is a protein that binds guanine nucleotides, such as GTP or GDP. So guanosine diphosphate, guanosine triphosphate. The binding of GDP versus GTP acts as an on and off switch, right, for this protein. So when I say on and off switch, what do I mean? When you bind something to a protein, right, that protein may have to change its shape to accommodate this thing being kind of stuck to it, right? And oftentimes, if a protein changes its three-dimensional shape, different surface features of that protein may appear or disappear, including three-dimensional features that serve as interaction sites for other proteins. So RAS is normally found associated on the against the cell membrane, right? So it's close to the surface of the cell, right on the underside. And RAS hangs out normally in a state where it's bound to GDP. So this is the off state of RAS. Okay. When RAS switches out the GDP and instead binds to GTP, right, the shape of RAS changes, and RAS then can begin to communicate with other proteins that might have still been floating by beforehand, but because RAS was not the right shape before, these proteins would just sort of bounce off of it and not care, right? But once RAS is bound to GTP, like guanosine triphosphate, its shape has now changed, and these proteins that float by now are the right shape to interact with it. And RAS then kicks off different signaling chains, different pathways of protein-protein interactions. You can see here, the three main ones are the RAS-RAF pathway that goes through these other intermediate proteins, MEC and ERK. There is also a RAL-GDS pathway, right? And there's also a PI3K pathway. So we will look at these, all right? So I'm just introducing these here. Eventually, these pathways activate certain transcription factor proteins in the nucleus, and those are the transcription factors that will then bind to the regulatory sites of certain genes, genes that do things like control apoptosis or cytoskeletal remodeling, or genes that regulate the cell cycle progression or cell proliferation and differentiation. So just by this set of different sort of phenotypic changes, you can already begin to appreciate why RAS is important, right? So RAS, again, is normally bound to GDP in an off state. GTP binding activates RAS. And so RAS kind of functions then as sort of like a supervisor that controls downstream workers. So we said in our analogy of the factory, the workers who actually have the skill to do little things like put together little pieces, parts of different machinery, right? They are the transcription factors. They work at a very low level, right at the level of the DNA itself, right? So these are the transcription factors down here. RAS is more like the floor supervisor who comes around, right? And sort of guides what the workers should be doing. You know, for example, telling the workers, okay, we need to kind of shift and do more of this. You need to speed up a little bit, or, you know, you need to slow down, something like that. Okay. I also want you to notice that the floor supervisor, this guy here with the yellow hard hat, right, he has to also be receiving instructions to know what to do, right? So if you notice here, there are these proteins, SHC, GRAB2, and SOS1, that are actually controlling RAS. They are the ones that determine whether the GDP gets swapped out for a GTP, right? And we will see how they are doing that as part of our discussion. But let's look at how this switch from GDP to GTP activates RAS. So here on the left is the off state of RAS. So this is just part of RAS. It's not the complete protein, but it's just the part that's relevant to our discussion, because this is where it binds to the guanosine nucleotide. So here's GDP, you can tell, because it has 2 phosphate groups in here. And when it's bound this way, this part of the RAS protein, so instead of showing it as a ribbon, this part they're showing as the actual atomic bonds, right, is shaped like this. If GTP is in there instead, you can see this has 3 phosphate groups. Well, I guess you can't really see the 3 phosphate groups, but take my word for it, there's 3 phosphate groups. All right, it causes RAS to kind of have to shift its shape just a little bit. These important regions over here, the switch 1 and switch 2, you don't have to really know that that's what they're called, but I want you to be able to see, right? Their position is slightly different, and that means the shape of RAS is slightly different now, and this is a surface that some of the other downstream proteins can now interact with, right? And that's how RAS then begins to coordinate those proteins to start doing their thing. So let's get back to this whole thing about this amino acid 12 being so important, right? Why do we see mutations in the vast majority of RAS mutations in tumors are this amino acid 12? Because here's the structure of wild type RAS here, right? So here again, these are in the blue, these are the switch regions that are shown above. Here is the GTP bound to it. Right, so this one you can really tell has 3 phosphate groups. Oh no, wait, here are the 3 phosphate groups up here, right? And so when it fits in there, right, normally GTP is temporarily held in there, right? The GTP then needs to then be converted into GDP, and the RAS will actually do that, right? So in a normal wild type RAS, this is the fit of GTP in there, right? And it's held, and then the conversion takes place so that RAS itself converts from the on state back to the off state, right? Just by catalyzing the cleaving of GTP back to GDP, right? If you look at these are 2 examples of real amino acid 12 mutations that have been seen in patients. So amino acid 12 is normally a glycine, but you can see one of the mutations here is it converts it to a aspartate, right? So a D. And what that does is it changes the shape of the pocket in which GTP is bound. So this is the actual amino acid 12 position here in orange, right? Another common one that's seen is at the 13th amino acid position, a G to a D amino acid substitution that also slightly changes the shape of the GTP binding pocket. In both of these cases, what happens is that this change in the shape of GTP binding pocket means the RAS hangs on to the GTP longer, and it does not convert the GTP back to GDP, right? So that means these mutations cause the RAS protein to just be stuck in the on position, right? Communicating with those different downstream pathways that start doing things like messing up the cell's ability to undergo apoptosis, the cell's ability to decide where in the cell cycle it should be, or the cell's ability to control whether or not it's just focused on proliferating. So here then, if we said that RAS is the supervisor regulating the workers, right, and SOS, GRAB2, and SHC are helping to control this by encouraging RAS to actually undergo the cycling between GDP and GTP, right? These guys over here themselves are being controlled by the big boss, right? So that's this guy here. He's the one who wears the suit and tie and only visits the factory, you know, like once every 4 months or something like that, right? So this is the boss over RAS. In this case, it is a membrane receptor known as EGFR, epidermal growth factor receptor. So it's a transmembrane receptor So part of it, right, sticks out of the cell. Then there's a hydrophobic transmembrane region that keeps it sort of anchored to the membrane. And then you have a cytoplasmic domain, right? I like the way that they kind of shaped it like these big nuclear missiles, as though somehow that's like indicative of how important it is. And these EGF receptors are very important in terms of many types of cancers. So when the cell receives messages from the outside that tell the cell, right, to change its behavior. For example, if this were a normal differentiated cell that's busy just doing its normal differentiated cell functions, right, making proteins that make it a muscle cell or a nerve cell or a gland cell. Remember, most of the time, a differentiated cell, that's what it wants to do. It just wants to carry out its normal functions. It doesn't want to bother dividing, right? It stays doing its normal differentiated functions because it's not told to do anything else other than that. However, if other tissues surrounding it are telling the cell, no, you should actually divide, right? Those cells communicate by sending little protein messages such as epidermal growth factor, or EGF. So these EGF molecules arrive at the surface of this target cell. They are bound by the EGFR receptor, right? And so the EGFR receptor then changes its shape. So suddenly it starts interacting with proteins in the cytoplasm, including things like SHC, that gets this starting to happen that then activates RAS, right? So epidermal growth factor in the surrounding media, if you have a culture of cells, right, and you put epidermal growth factor in it, they will be received by the cells by using their EGFRs, and they will then activate RAS. And RAS then will do things like it'll start pushing the cell through its cell cycle, right? It'll then encourage the cell to proliferate, right? We will also look at this pathway here that's mediated by EGFR, the JAK-STAT pathway, which is important for cell survival. So what's an important take-home message here, right? These oncogenes or proto-oncogenes like RAS, right, they are normally present in our cells, right? That's the big point we were trying to make before, that even though you think of them as these terrible oncogenes that drive cancer, right, the reality is they play a normal role. And that normal role is they're hanging out, waiting to interpret signals from the outside that then encourage the cell to divide, right, and proliferate. Those signals that come from the outside, right, need to be received by the big boss, the receptors that actually take that message and start talking to things like RAS to direct them around. And you can also see then that if you have mutations in RAS that cause RAS to no longer care about waiting for these messages from the outside, and the RAS just activates all the time, then you can see how that would lead to the cell starting to proliferate out of control. Right. So many mutations not only happen in things like RAS, but of course they can happen in the boss proteins themselves, those EGFR receptors, right? Because they are just proteins. They are just coded for by the EGFR genes. Those EGFR genes can get mutations as well that result in misshapen or defective EGFR receptors. Here's two examples. Let's say you have a normal wild type EGFR here, and this is how it's supposed to look, right? You have the exterior domain that's going to bind EGFR, you have a transmembrane region, and then you have the cytoplasmic domain, right? Normally what happens is when epidermal growth factor comes and binds, when it changes the shape of EGFR, it causes two EGFR molecules to come together and form a dimer. And it's when it's the dimer, then the cytoplasmic part of the two halves, that's what forms the right shape now for other proteins to start associating with it. Let's say you have a mutation that is a missense mutation. Or no, not a missense, but a nonsense mutation, right? That, wait, I forget now. What is it that would cause a premature truncation? That's missense, right? Well, anyhow, so let's say it causes the gene to now have a stop codon in it where it shouldn't, right? And as a result, the protein that gets made from this prematurely truncated gene is itself a version of the EGFR that is truncated. It no longer has the rest of the protein. In this case, it might be that this causes then this truncated form of EGFR to automatically just dimerize with other truncated forms of EGFR, right? Thereby activating the cytoplasmic region and constitutively or always then being on, right? So mutations in these types of membrane receptors are also very important for potentially causing things like cancer. Right. So that was RAS, and we talked a little about EGFR. Let's talk about Src, right? Because Src is the first oncogene we started talking about this semester. So this is what Src looks like. So Src, here's human Src, the normal version that's in our bodies. And here's chicken Src, right? The one that, since we talked about the story of Peyton Rouse and the chicken, not surprisingly, evolutionarily, it's very conserved. So they're all it's pretty much the same protein. I think the one is maybe a few amino acids, different than the other in parts that don't matter. Right. But the parts that do matter are these different domains. So the amino acid sequences that are sort of shaded in gray, green, purple, and blue here, right? These are the SH domains, okay, that are conserved, not only in Src, but many other proteins also utilize these types of domains. Right? But they were first discovered in Src. And so that's why SH domain means Src homology domain, right? So in other proteins, that we evolved to use this sequence in other proteins because the type of domain that's made by them in a three-dimensional protein is very useful, right? Because that's how Mother Nature works evolutionarily. If you come up with a particular protein structure that's very useful for something, well, it can be part of other proteins as well. So many other proteins have these SH domains in them, but they were originally found in Src. All right. So what you see here is in this cartoon on the bottom, right? These are the domains represented in a very simple sort of geometrical shape. So the N-terminus, the M domain, where the SH4 domain is, keeps Src also anchored to the underside of the cell membrane, sort of like RAS is also hanging out there as well. Then the SH3 domains, the SH2 domain, the SH1 domain, it folds up in this way. So the SH1 domain is the kinase domain, right? Src functions as a kinase, meaning its job is to transfer phosphate groups from one protein to another protein, and in doing so, encouraging target proteins to change their shape and activate once they have a phosphate group stuck to them. Normally, Src is held in this sort of inactive state here, where the domains are kind of wrapped up on each other. It's held this way because the very C-terminus of it, over here, this yellow bit that kind of wraps around, right? So this is the very C-terminal end of the protein. There's a tyrosine on here, tyrosine 530 in humans, that gets phosphorylated. And that phosphorylated tyrosine sticks very tightly to the SH2 domain and keeps the whole protein kind of folded up this way, inactive, right? If something comes along and removes this phosphate from 530, then what happens is the protein unravels, right? And when it unravels, then that kinase domain, the SH1 domain, is now available to do its thing, right? You can see the tail, the C-terminal tail that contains the 530 now is just kind of sticking up out here, right? Because it's no longer phosphorylated, all right? Here is the structure of Src a little bit more realistically here. You can see here are the different domains that correspond to this cartoon over here. And here you can see, here's the C domain, right? That's wrapped around and is tightly associated with the SH2 domain. And this keeps the protein in an inactive state. All right. So let's look at something like the chicken Src, right? We said the researchers who were first looking at Rouse sarcoma virus, they identified that the virus, right, had something kind of different going on in it, right? And that something different was a different, well, the Rouse sarcoma virus had that extra bit that was the Src that got taken by the virus at some point in evolution from the chicken genome, right? And now was part of the viral genome. And then over time, right, it mutated It mutated in such a way such that when this virus reinfects chicken cells, right, this version of Src that now gets expressed inside the chicken cells is oncogenic. So if this is the normal version of Src that was maybe originally taken from the chicken genome, and this is the version that would still be present in the normal chicken genome, what do you think the viral oncogene form of VSrc looks like? Based upon what we've been talking about down here of how the Src is normally regulated. Okay, again, I would love it if one of you just kind of threw some ideas out there. What do you think the viral version that mutated in the Rous sarcoma virus looks like compared to this one up here? Did everybody go to sleep? Anybody throw out a guess? If the Src is normally tightly controlled by its own C-terminal domain wrapping around over here, right? And the C-terminal domain normally gets deactivated, that opens the Src up. Right? What do you think a mutation might be that could cause this to become oncogenic?
There could be a mutation at the tyrosine 530, so there's no phosphorylation binding, so it's constitutively activated.
Yeah. Okay, so you might have a point mutation that results in the removal of this tyrosine, and it changes it to something that doesn't get phosphorylated. But even more dramatically, what if those residues were just no longer there in the first place, right? So in fact, so you're on the right target, you're on the right track, and certainly such mutations probably exist. But in the classical case of the chicken VSrc, this is what chicken VSrc looks like, right? It is truncated. So the mutation, just like what we talked about with the EGFR receptor, it's truncated, so part of the protein is missing. And in this case, the C-terminal domain is no longer there. So the VSrc form of the protein, when the virus introduces it into a host cell, and the host cell is hijacked and starts making this truncated form of Src, right? There's no control over the Src anymore. And Src just starts going crazy because it's always on inside of the cell. Okay. Another example of how genes can have different mutations are through amplifications, right? So different genes that normally need to be kept very tightly controlled, not in terms of the protein function, right? Because what we've been looking at so far this lecture is if you actually have an altered protein structure that somehow removes or eliminates an on and off switch, right? Whether you're talking about the RAS, eliminating its on and off switch so it doesn't properly switch between GTP and GDP, or the EGFR receptor, if you remove part of the protein that serves as the on and off switch, because that's the part that needs to normally bind epidermal growth factors, right? You take that switch off and the rest of the receptor might just activate by itself, or the Src, where you remove the C-terminal part of the protein so that the Src is always unraveled and it's always functioning. Another way that mutations can mess up the function of proteins is through overamplification, right? So here are some examples. There are examples in certain diseases where the RAS is just amplified. Okay, so you have different mutations that actually cause the expression levels of these critical proto-oncogenes to be elevated beyond the point where the cell can properly control them. Another one is Myc, right? And so that's what we'll end up talking about today. And Myc is another important oncogene. And you see that the version of Myc we're going to talk about is c-Myc. So this is the original Myc that was described. And it is found on chromosome 8q24. Right? And you can see that up to half of all carcinomas contain some sort of a Myc mutation. And in most of these, it's actually not a mutation in the Myc itself, the gene, but a mutation somewhere that impacts the expression level of Myc. You can see that the type of protein that Myc is itself a transcription factor, right? So previously I said, oh, the important proto-oncogenes are the supervisors that actually control the transcription factors, right? That's true for many types of oncogenes. Myc is a notable exception, where Myc itself is a transcription factor. And the reason why Myc is so important as a potent oncogene if it goes bad is because it is a transcription factor that's used across the board, right? It has the ability to control a large majority of the genes in the genome. So if something happens to Myc and the levels of Myc go out of control, the cell has a hard time keeping tabs on proper expression level. So just to show how important Myc is, here's another Kaplan-Meier plot, right? Remember, Kaplan-Meier plots are these clinical data sets that are a nice way of showing how following a group of patients from a starting point to an end point, whatever that end point is that you decide to call the end point, you can follow two maybe closely related groups of patients who differ by some kind of diagnostic variable, right, or prognostic variable. So in this case, they are following pediatric neuroblastoma patients, right? And they were looking for whether or not these poor kids who first present with neuroblastoma and they show up in the clinic and they start getting treated, right? And they take some biopsies of their cells, right? They're broken into two groups. Those kids who had fewer than 10 copies of Myc present in their genome, right? So this actually shows you how sensitive Myc is, or how sensitive the normal cell functioning is to Myc, that you're not just saying 1 or 2 copies, right? That in reality, a large group of these patients, right, have fewer than 10 copies of Myc, and that's actually a suitable sort of cutoff for comparing. So fewer than 10 copies of Myc in the genome versus greater than 10 copies of Myc, right? And you can see very clearly, right, those patients who present with tumors and their tumors already show more than 10 copies of Myc, right? They have a far poorer prognosis, right? So on the x-axis is years after diagnosis, right, before this particular endpoint data was collected. And this endpoint is EFS probability, which means event-free survival, right? So it's kind of a general way of capturing any kind of recurrence of symptoms, right? Any symptoms that crop up, right, that would be considered a new event that has happened as you're following these patients, right? So Myc very clearly has an important role here. So the story of Myc kind of ties in with a story of a particular type of cancer known as Burkitt's lymphoma. So in the mid-1950s, Dennis Burkitt, who was a physician who was practicing in sub-Saharan Africa, right? So he was trying to bring sort of westernized medicine to many of these villages that are sort of cut off from modern medical procedures. You know, the clinics are very poorly sort of supplied there, not a lot of resources. So Dennis Burkitt was dedicating himself to working in Africa. And he observed that there were an unusual numbers of incidences of pediatric facial tumors in kids in these villages. And what he began to do is he began to map out different villages that he visited where he would see these kids with facial tumors. And this is the map, right? And you can see over time, it looked like this map was sort of focused on the central belt of Africa, with some cases down in the southern tip. Right? So he published this, but you know, at the time, nobody really cared, right? Because he had no explanation for it. It was just this curious finding that these kids in these villages in sub-Saharan Africa, right, were developing these facial tumors. So one day, the story goes, you know, Burkitt is giving a seminar about this. I think he's visiting Europe and he's giving this seminar, and attending one of his seminars is this guy, Michael Epstein, right? So Michael Epstein, right? And so he took a real interest in this because he noted that the distribution on the African map of these cases of tumors corresponded very well to what Epstein knew was sort of the distribution of things like mosquito-transmitted malaria, right? And Epstein was very interested as a virologist in things like, the viral transmission through things like malaria, and not through malaria, but through things like mosquitoes. So he took quite an interest in finding out whether it could be that the that the Burkitt's tumors that he was seeing could somehow be linked to a virus, right? So Epstein and his graduate student, Yvonne Barr, then collaborated with Burkitt. And Burkitt would send these tissue samples of these tumors, from Africa all the way back to England. And so, of course, back then, this is the 50s and 60s, they didn't have FedEx overnight, right, with like dry ice packaging and stuff like that. It would often take many days, a combination of boats and airplanes. And by the time, the samples arrived, the Epstein and Barr would rush down to the airport, get the samples, rush them back, and try in their lab to kind of under the microscope see if there were virus particles. And they couldn't do it because what they realized is that these tumors, right, were very sort of dense and fibrous. It was very hard for them to get the cells to kind of like lyse and release the contents. And they didn't even know what they were looking for. They hoped to see a virus. So one day, though, one of the shipments from Africa got delayed because of bad weather. And the plane arrived in England, but it had to sit, I think, on the tarmac or whatever for several days, right? And so Epstein and Barr, by the time they got the samples, they were just nasty, right? Because they had gone all yucky and putrid. But they said, well, these are valuable. We can't throw these away. Let's just go ahead and see what we can do. And what they realized is because the tissue had gotten all nasty and putrid, the cells actually lysed much easier. And it was from that sample that they were able to actually identify the virus particle. And so these are the virus particles in a cell infected with this virus that then became known as Epstein-Barr virus, right, named after the discoverers. So it's interesting to think that this was a discovery that just happened because of chance, because of bad weather that delayed their ability to get to these samples. All right. So this Epstein-Barr virus, what's the relationship with developing these lymphomas that were seen in the kids, right? So in the kids, these lymphomas were later named Burkitt's lymphomas. When people later analyzed the karyotypes or the genomes of these kids, what they saw was that a translocation event had happened. A translocation is when one part of one chromosome swaps with, let's say, an arm of another chromosome. That's how you get the multicolored sort of candy cane looking pattern that we see in those really messed up cancer karyotypes, right? Huge chunks of one chromosome have swapped around. That's a translocation. In this case, what they saw was that a part of chromosome 8 had swapped with a part of chromosome 14. So the notation that we use, this is typical notation here. When you're talking about a translocation, is you use lowercase t and then in parentheses, right? So this is a translocation between chromosome 8, semicolon, and chromosome 14, right? So that's how you describe this. So in a majority of Burkitt's lymphomas cases, this T814 translocation has taken place. So clearly the important thing is what genes are actually on these parts that are being swapped around, right? On a normal chromosome, Myc is at 8q24, right here in red, right? On chromosome 14 at q32 is the IgH gene. This is the immunoglobulin heavy chain gene. For those of you who have taken immunology with me, right, this is the gene that codes for the heavy chain part of immunoglobulin molecules that go on to form antibodies. Remember, an immunoglobulin has 2 heavy chains and 2 light chains. That's the Y shape of an antibody. So these are swapped around in cases of Burkitt's lymphoma. And unfortunately, what has happened here is the intact Myc gene, right, has gotten stuck behind the promoter of the IgH gene. So here on the right, you can see here, normal chromosome 14, you have the whole IGH gene. So it has a promoter in front of it that drives the expression of the gene. So in certain cells, immune cells, such as B cells, right? B cells would be the only cells in your body that bother expressing this particular gene to make heavy chain, to make things like immunoglobulins and antibodies. Remember, every cell in your body that has a nucleus has these genes. It's just the other cells don't bother actually expressing product from these genes. But in B cells, right, you're constantly making heavy chain. That means the promoter here is a very sort of like, how should I say it? Very active, constantly constitutively on kind of promoter, driving production of Ig heavy chain. On normal chromosome 8, where MYC would normally be, its promoter, right, isn't like that. It's much more tightly controlled. So your cell tightly controls how much MYC is actually made from it. But in the T814 translocation, the MYC, I said intact MYC. It's not fully intact. It's basically the functional exons of MYC, right, have gotten now stuck right behind IGH. So the overactive, constantly on Ig heavy chain promoter is now improperly constantly driving expression of MYC. And in that way, MYC suddenly becomes an overwhelming oncogenic signaling in the cell. And again, not all cells, right? Because not all cells would care that MYC is stuck behind the Ig heavy chain. Because if this happened in something like a muscle cell or a hair cell or a nerve cell, right, and this exact same thing happened, there wouldn't be much consequences, because in those cells, the Ig heavy chain promoter is shut off anyhow, right? That's interesting to think about, right? But if it happens to be a B cell that normally has its Ig heavy chain on, then those B cells can start transforming. And that's what gives rise to lymphoma, right? So very specific type of cancer involving these lymphocytes, these B cells, right? Because they use this particular locus. And if the translocation happens in them, that will cause MYC to be overexpressed. So what is the tie-in with malaria, right? So this is where the story gets a little funny, because it turns out that malaria has nothing to do with it. You know, it's an important part of the study, because that's why Epstein first got interested in it, because he was already aware of malaria, right? And he was interested in these mosquito-borne vectors of disease and things like that. But it just so happens that there's evidence suggesting that people who get malaria, for some reason, the malaria, and this is still not understood yet, it increases the severity of Epstein-Barr virus infection. Right? So even though the mechanism of Burkitt's lymphoma very much is just Epstein-Barr virus causing these crazy translocations to happen, leading to the MYC 814 translocation, right? The additional presence of malaria seems to increase that. And we still don't know why. So that's a fascinating question, right? So it's not quite coincidence then that this is happening where you see malaria, but it turns out the malaria itself is not the cause. All right. So here are some other examples of other translocation events that lead to specific tumor phenotypes. So we already talked about MYC. If MYC is involved in the T814 translocation, it leads to Burkitt's lymphoma, as well as other types of lymphocyte-specific cancers. There's some other ones here. Let's take a look at this example here, the BCL. All right. Oh, right. So I should point out that translocations resulting in deregulation of known proto-oncogenes, such as MYC, those are the most frequent types of translocations that you see in cancers, right? Again, just like with the whole, why do you see that like 99% of RAS mutations in tumors have a mutation at amino acid 12? It's because you already biased what you're looking at, because you're starting with the tumors, right? Similarly, you're starting with tumors and saying, what types of translocation events, do these tumors seem to have, right? The majority of the events that you do see that took place result in deregulation of known proto-oncogenes. Okay. So deregulation doesn't necessarily involve increased transcription levels of a gene then. There are other ways that you can have funny translocation events that alter loss of regulation. So a very interesting one is this, what we call BCR-ABL translocation. So different translocations might result in part of one protein genetic sequence being fused, right, introduced right up against the protein sequence of another protein, or the nucleotide sequence that codes for another protein. And as a result, if this resulting gene, which is a hybrid, right, of two genes put together, if it gets transcribed and translated, you might have what we call a chimeric protein, right? A protein that's actually parts of two proteins as one. Remember, a chimera in, what is it, Greek mythology is the, what is it? It's the lion's head on the sheep's body or something, or I don't know what it is, right? But so that's what we mean by chimeric, right? It's two parts that don't belong together that normally are. So this T922 translocation is one of these. It involves a part of chromosome 22 that contains this gene BCR, and a part of chromosome 9Q34, that contains this gene called ABL. And when this translocation happens, you get a hybrid protein that's made here, because the resulting fusion still successfully is able to be transcribed and translated. And the protein that's created is this BCR-ABL protein. All right, so this is the last slide for today. Right. So what is ABL? So ABL is a tyrosine kinase, just like Src. Right? It's not quite clear what types of kinase targets ABL has in the cell. It's pretty clear, though, that normally the normal function of ABL is not necessarily related to things like cell proliferation or division. So really, normal ABL is not considered a proto-oncogene, because the function of ABL normally has something else to do with other pathways not related to cell cycle, right? But when it's fused to this BCR part, right, the BCR gene is a bit of a mystery. We still don't know what the BCR protein does. But what we do know is that the BCR protein tends to go to different parts of the cell, right? Parts of the cell that, importantly, the normal ABL protein does not go to. But if you have this fusion that takes place, what ends up happening is the BCR part of this chimeric protein then takes the ABL part to other parts of the cell that the ABL should not be going to, has no business going to. And what we think is happening in those regions, ABL is then to start interacting with different downstream targets to do its kinase function that normally it would not have a chance to interact with. And those pathways are things like cell cycle progression pathways. Okay. Okay, and just to make this note here, I show you the structure here because this is ABL, right? And you can see it has these SH2, SH3 domains as well, and a kinase domain. If you compare it to Src, you can see that ABL is very similar to the structure of Src, right? So just like Src has very strong proto-oncogenic, potentially oncogenic consequences if it improperly activates, that's because Src is already in a place in the cell where it's ready to interact with those cell cycle pathways. ABL normally is not found there, right? But if ABL is brought there because it's half of it is fused to BCR and BCR wants to go there, right? Then suddenly ABL can start also contributing to the activation of those pathways, right? Kind of cool, right? I think it's kind of cool because it shows you, right, that these proteins, right, they're ready to do stuff. And a lot of times it just depends where your cell carefully keeps them sequestered in different parts of the cell to limit what they are or are not allowed to carry out their function. Right? Okay, let's stop there. Okay. Okay.